All right, so let's start with today's webinar. So hello everyone, a very warm greeting to one and all. My name is Srigauri Krishnakumar and I would like to welcome you all for today's webinar on Introduction to Bioinformatics in our program. As biology is saturated with complex data sets that have to be sorted, explored, and looked into, anyone handling data generation, analysis, or decision making based on data has to gain some level of data science skills. In most biological and biomedical settings, you will be expected to run or implement programs written in Python or R. Our programming offers a complete range of functionality that is, you can leverage to perform in-depth statistical analysis, visualization, and annotation. Getting started can be hard. Programming is like learning a new language. And that is why we offer easy to follow, structured, and simple coding tutorials designed around bioinformatic challenges with mentor guidance in this upcoming introduction to bioinformatics in our program that will commence on December 1st. And the aim of today's webinar is to just introduce the participants to program details such as program mentor, program curriculum overview, course structure, expectations, deadlines, intended outcomes, and program registrations. So before we begin with today's webinar, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of the program, students will have access to the omics logic training that has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine program logistics and adapting them to the needs of students and researchers around the world. And this training has been completed by participants in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. And students will also have access to the omics logic resources. But how does it help advance bioinformatics teaching? Omics Logic is a portal for practical and theoretical learning of bioinformatics. It takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give the student a clear path up the Bloom's taxonomy pyramid. And no matter what the user's initial experience with bioinformatics is, Omics Logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large datasets, and introduction to basic coding languages like R and Python, or even beginning a data analysis project. And this also comes with an access to the Omics Logic courses, which includes courses on bioinformatics, precision medicine, multi-omics, that includes genomics, transcriptomics, metagenomics, and epigenomics, data science that includes using R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. And users will be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. And the platform includes demo pipeline, as well as data management and analysis cloud infrastructure to run the bioinformatic pipelines. And different stages of analysis are performed in different sections on this multi-omics platform. And now to view the various courses and example projects, you need to first sign up on the Omics Logic Learn portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features that you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles. This could be your Facebook, LinkedIn, Instagram, OrcID, or even Twitter. A brief bio about yourself that will consist of your educational background and research interest. And the mentors can view your progress under the Activity tab, and the courses you are currently completing will appear under the Courses tab. Finally, after the completion of your course, the certificates will appear under the Certificates tab. So with that, now let me walk you through a brief demo on how you can sign up on the portal. So kindly make sure to visit the link and sign up along with me so that you can get access to all of the resources that we will be discussing during today's program. Okay, so those of you who are new to this portal and who do not have an account, here is a demo on how you can create one. So for that, just click on the option that says create an account that you see here. And here you can enter your name, give an email ID, and enter a password. Or you also have the option to sign up using any of your social media handles. This could be your Google, Facebook, Apple account, GitHub account, or even Twitter. So with that, I'll just pause for a couple of minutes so that if the participants have any queries on signing up, they can let me know. And for those participants who have already signed up, you can just click on the login now option that you see here and just simply click on the sign in. So I'll just wait for a couple of minutes. So once you have done signing up on the portal, you can leave a done in the chat box. And if you already have an account on the portal, you can just leave a yes so that I can get a fair count of those who already have a pre-registered account on the portal. If you're facing any technical issues, you can mention that as well. All right, so we have one participant who has already signed up, and I'm waiting for the rest of the 50 plus participants to sign up. Today, we would like to make it more interactive. So to get access to all of the courses and program structure, it will be really great if you could create an account so that we can go along and learn about what are the various topics that will be covered. So I'm just going to click on sign in since I already have an account on the portal. All right, so once you sign in, you'll be able to see the various courses and example projects that are there on the portal. So the topics range from oncology, infectious diseases, to machine learning courses as well. All right. And so for instance, if you're interested to learn about R, you can just simply enter the keyword R, and it will show up all the courses and example projects that makes use of R programming language. And let's say you have another field of interest. Suppose, let's say it's oncology. Then you can simply enter the term oncology or cancer, and that will also show you all the courses and example projects that will help you learn the topic. All right, so with that, let me walk you through the next important part about setting up your profile. For that, click on the tab that says welcome back with your email ID. And once you click here, you'll be able to see your profile. And how do you edit the profile? For that, you can just click on the button that says profile. And here, make sure to upload your profile image. And your name has to be in proper capitalization. And you can also link any of your social media handles. For instance, I've linked my LinkedIn account and my org ID. And also make sure to add your bio. That is, it will consist of your educational background and research interest. And once you have done, you have updated your profile. Now, let us also explore some of the various tabs that are there in the portal. That is, under the activity tab, as I mentioned, the community manager and the mentor of the program will be tracking your learning progress. And to view the various courses that you're currently taking, it will be shown under the courses tab. For instance, currently I'm taking the effect of non-antibiotics and Rett syndrome example projects. And once you complete a research project as a part of the research fellowship program, you'll be able to upload it under the projects tab. And if you're enrolling for the program, for instance, the introduction to bioinformatics in our program, the program progress will be appeared under the programs tab. And finally, after completing the assignments and the required quizzes, the course certificates tabs will be appearing under the certificates tab. So with that, I'll just pause for a couple of minutes. And if the participants have no queries, we'll be good to start with today's session. So are we good to go? Is that a yes or a no? Is anyone facing any queries? All right, so I'm just going to pass on the stage to Dr. Raghavendran Lakshmi Narayan to start with today's session. He'll be giving you an overview of the program structure and the various topics covered. Followed by that, we'll be discussing the program registration cost as well. So along the way, if you're having any queries, all right, so we have a question from Muskan. Can we upload the certificate on our LinkedIn profile after completion of the course? Yes, absolutely. I have uploaded it as well. and it helps me network with more people on LinkedIn. So that's a yes. So if the participants have any other queries, please let me know. Along the way, you can share your queries as well, and we'll be happy to answer your queries towards the end as well. So with that, the stage is all yours, Dr. Raghav.
Thank you, Sri Gauri. Hello, everybody. Welcome to today's session. So let's have a hands-on session, and let me set up my screen share. So quickly, can you type in the chat box, how proficient are you in any coding environment? So 0 being absolute novice, and 10 or above 8 or 7 being, you will be able to get codes from publicly available resources like GitHub and Stack Overflow, and then modify that to your needs. So please let me know. A beginner would be something like 1, 2, and moderate experience would be like 5. Okay, excellent. So let me find this. Thank you. Thank you. Let this flow. Let me set it up. Okay. Thank you. Thank you very much. Thank you, everybody. So, okay, we will take this real slow, and we will perform analysis that will help you understand how we could process and how we could use R to perform RNA-seq data analysis or transcriptomics analysis. Let's see. We're going to evaluate or cater data from publicly available resources and get. Let me set it up. My pardon me for a moment. Let me rejoin because my Zoom is not looking at my screen. Okay. Still loading. I think it's starting to. Okay, now it's fine. Can you see my screen? What do you see? Do you see the program page?
Yes, so we can see your program page.
Okay, thank you very much. Okay, thank you very much. So, yes, thank you. Let me set this up. So I have opened a chat next to you, next to me, so that I can follow your responses in chat. So we will start with, for example, absolute basics of getting started with bioinformatics in R. So this associated resources will actually help you load, for example, the data in R, and it'll help you understand the syntaxes. Let's take a look at one of the resources. That's okay. Data processing and visualization in R. Let's go there. So I'll just quickly tell you how the coding resources are structured, and then we will start with some demonstration, some analysis. Let's see. So this is. Yeah, so this is how it is structured. First, we start with introducing you to what is the aim, or what is the problem statement that we have. For example, in this lesson, we are going to address different data processing and data visualization options that are available in R to visualize any structured data. So, so first, you will have options to learn the code, or you will get where you will understand in blocks of codes the exact code commented properly, and around the code we will give you explanations of what each and every components of this R code is going to tell you. Right? For example, in this simple code where we are going to load a table into an environment like R. So first we have to use a function that tells R to do what we want to do, like load the table, and we have to give this function the location of where you will find the table. And this location can be from your computer, and for ease of access of the data, we have uploaded a lot of these curated data sets to our data sharing platform, and then we will give you those links so that you can follow along as we go in the analysis. And then what follows is the characteristics of your data. I will come a little bit more detail into what these arguments are in a short while. So to explain you what is happening, so this is a code, and what you see around this is going to be different parts of the code explained and their typical output actually given, right? For example, if you are going to plot something, we will give you what to look for if you are going to use this plot or use this code to plot a data set. So we will start something like loading the data into our environment, and then explore the data or process the data in a necessary format so that we can apply some statistical analysis to this data, right? And this will include, for example, remove some of the duplicates, and this will include, for example, remove some of the not applicable values, and you will get everything in more detail, and how this affects the data and how that increases or improves accuracy. We will get to that in more detail. And then we will explore again, this talks about some transformation in the data scale, like logarithmic transformation. You will also learn about why is it necessary, how is it useful, and how to do that. So, and finally, you will visualize the data, this simple visualization, which plots the data in a box plot format to understand the data distribution across different samples. So you will get more and more explanations on how to perform not only this analysis and other data visualization options, and how those visualizations are going to help us understand the patterns of the data. How, for example, you can learn a little bit about the data distribution in box plot, and what will you learn about, for example, say in scatter plot, and etc. So when you keep exploring more and more, like you can come to heat map and histograms and density plots, etc., etc. So there is a myriad of different exploratory data analysis that can be performed, and you will get a lot of examples in how to do this analysis, and how is it going to help us understand the data and their pattern generally in the omics data type. And then if you scroll down, you will, for example, perform based on the understanding of the exploratory data analysis. You can set up comparisons for the statistical tests to evaluate our data patterns. And that is what we will do a little bit in a short while. And then followed by, you will have some introductions to what if you need advanced statistical analysis like machine learning, and how to use, for example, metagenomics data, how to make use of metagenomics data, which comes up with its own challenges and opportunities to understand a little bit more about data. So we will, for example, in this whole curriculum, we will start with working with sequences and covering some genomic variants in R, and then we will where we are going to address how to perform or how to use R's tools to understand patterns in the genomics data analysis output, like variant calling output, and how to identify the significance of those variants. And then followed by you will perform, for example, downstream analysis on transcriptomics data, which you will do it today. And then where we are going to cover, for example, starting from obtaining data from publicly available resources, like we take a take a paper and then find the data, and then take the data in the necessary format, and then start performing this analysis, right? And then over the course, we will also discuss the pluses and minuses of a couple of analysis, statistical analysis options. Okay, and then we will follow this up with what if straightforward or normal statistical analysis is not sufficient for your data analysis. In that case, we have to use advanced statistical methods like machine learning to understand the patterns in your data. So this will also be covered in the resources, and then we are going to finish or end the program with analyzing or using R's tools to understand metagenomics data, which is a little bit different from the other forms of data, as it actually covers or as it contains information from multiple different entities like microbes instead of one cell or an averaged out cellular expression or genomic information from the cells that we usually deal with in the genomics and transcriptomics data. So let's start with, for example, setting up some analysis in Google Colab. So we will use Google Colab's environment for today's analysis. So if you have if you do not have any introduction to Google's Colab, Google's Colab would be, for example, let me get the link. So all of you, whoever has Google support or ID can use this collaboratory notebook. So you will open up a page like this. So, okay, can you click on this specific link and then open up a page like this? This is Google allocated resources for you to perform some simple calculations in R and Python separately, and use the shareability of this Google's notebook to share it with your peers, with your friends, with your collaborators, etc., so that you can use the collabs and their tools effectively. So that's what we are going to do today also. So if you see this such a screen, and you can actually change the name of this collab here, my Domo collab, and then you have to connect to the Google's hosted services or resources so that we can run some codes in this collaboratory environment. So we will simply press connect or do something like connect to a hosted runtime here. So all of you, can you connect? If you connect, it's going to show something like this. It's going to show you something like this until it's going to take a while, and then it's going to show you a green tick and then some associated resources that you are not resources, actually hosted runtimes, right? Host runtime that you will gain access to. Can you press this connect and then wait for this connection to work out? So please give a 1 or yes in the chat box when you see when you observe a green tick mark next to this connect. So we will discuss about what is the data that we are going to discuss and that we are going to analyze today. But before that, we have to start setting up some of the libraries and start installing some of the modules and libraries. So we will let that install and then come back and discuss the data that we are going to discuss, and data that we are going to focus on today. Okay, looks like, yeah, good, very good, excellent. So now, now comes the little bit coding part. So we have to install these libraries. So I'm going to copy this and paste it onto your chat box. So I'm going to copy this and paste it in the chat box. So I'll paste it here. You copy this and paste it here, right in this code. You see this code? If this code is not activated, double click in this one and paste it here. So do you see a screen like what I have obtained? And then once you have this one ready, you can execute this by pressing this run cell option on this play area or in this specific button. So after you press that, it's going to keep installing for a while. You will see some outputs in this section below, but let it run. So just let me know if everybody is able to start installing these packages that we are going to need to use this. So give me a response in chat. So it will install something like easily from 2 minutes to 4 minutes, depending upon, maybe many of us are using, it might take a little bit longer, so that we will use the time to discuss the data. But yeah, we will do this here. Okay, yes, some more yeses. Is everybody here? Is it running? Is it working, or is anybody getting a problem? So this is absolutely necessary step for us. Installing, very good, not yet. Okay, Alpana Sharma, I think you did not, wait. For those who are getting error like Ruth and Alpana Sharma, I think you are actually installing this in no paste option there. Use Command V or Control V from your computer. So Google doesn't work very well with right click and paste. So you have to use your keyboard shortcuts to paste. And Ruth and everybody, please use, for example, this link to open up a collab, and don't use any other link, because you are trying to make an R script run inside a Python notebook. So that is why you are getting an error. Use this script to run. For both of them, you will get the same extension. So use this link to open up the code, and then still installing. It will install like 3 to 4 minutes. So is Ruth and Alpana Sharma there? Please open this up, open the collab with this link, and then paste it and start installing. Are you there? Let me know. Yes, very good. Thank you, Ruth. And what about Alpana? Yes, okay. Okay, so let's look for the data set. The data set that we are going to analyze today is from this publication, okay? Okay, installing. Yeah, it is going to install for 2 or 3 minutes. So let's focus on this, the data set that we are going to understand today. So it's going to be this data set where we are going to follow along the data that is present in this publication to understand genes that are that can be identified from differential gene expression analysis using something like comparing colorectal cancer patients with normal patients. And that's what we are going to do today. So this, let me copy this publication. So from most of these publications, either in PubMed link or any other link, you can come to the data part, right? And then get a link to where the data is available for this publication, right? So you can read more about the publication, what they actually identify based on this, right? Yeah. So let's go down. Yeah. So they do something like they compare, for example, gene expression data from colorectal cancer compared to the normal cancer, and then gene expression data from metastasized colorectal cancer compared to the normal, and also compared to the cancer cells, and try to follow along the risk factors or risky genes whose expression might be correlated with the metastatic stage of the cancer. So that's what they actually do. That involves a lot of other analysis like survivability analysis and etc. So this is an excellent paper to go through to understand how transcriptomics data can actually help us characterize the genes based on their involvement in tumor progression, right? And if you come down, for example, in the data page, right, it's also available here in the supplementary information, but in the data page, you will get a link to the gene expression omnibus data page where you click this and it's going to go to gene expression omnibus. Let me come back. It's already, I have used it here. So this is where usually we look for data, but coming from the publication is another way of getting to the data. So this is the repository in NCBI, which hosts many transcriptomics data and other omics data type. So here you can read a little bit about the data, a little bit about this overall design, a little bit about the what was studied, and you will see something like this. You can get how many samples has been studied, and more importantly, and what are these samples and their sample annotations, right? We have something like 23 primary cancer tissue, and then we have something like 24 normal cancer tissue, and then we have 20-odd metastasized cancer tissue and their gene expression data studied using high-throughput sequencing methods. So this page also has the input data, or not in the processed data, in supplementary file. So you can download this using this HTTP link, or the sample characteristics can also be explored from series matrix file. So usually this is how we access data, but only for few of the studies they are going to give an alternative data, published data link. Like this one has published their data in NCBI and also in EBI links, right? You should explore both of them. Why? Because when you download this specific data, right, and this data is going to open up like this, and where you will have each and you'll have the expression levels quantified for each and every one of the samples in a separate file. So you will need something like you'll need to load up all of these files into environment like R to merge them into one big table. Usually that's what we get when you perform a transcriptomics sequence analysis or RNA-seq pipeline analysis in the big data bioinfo server like Taubot bioinfo server, right? But NCBI data has the gene expression quantified for each and every sample separately. This is useful, not to say that this is not useful, but for our purpose, we will need one big table that contains the expression data for each one of the samples tabulated accordingly. So that is, and again, if you download the series matrix file, which is going to, for example, give you the characteristics of the sample. Yeah, okay. Okay, so how is your installing going on? It's still going on? Has anybody got their installation complete? Maybe many of us are using, so it's going to take a while. Still going on, yes. Ongoing, okay. Anybody managed to complete this? Ongoing, for many of you ongoing on BC server, okay, the server BC. Now I think many of us we are starting to ongoing, showing cannot open file. You have not even opened file, no? Which link are you using, Arjun? Same link, okay, can you paste it? the recording will be shared, and yeah, we are waiting for the installations to complete. I'm being very slow, or I can even slow down. So, okay, let it go through. So we will come back to this, right? Okay, let's discuss a little bit more about how to get the data, right? So this is a series matrix file, which is going to a link for, okay. This is the link with which you can open up an R. I think now the server has gotten too, okay, server has gotten too busy. Yeah, maybe some of you can follow along. Let's see. Anybody managed to complete installation? That's why I thought we would finish up the installation and then discuss the data download. It's going to take some time. Still installing. Okay, it's been a while. Okay, so this is how you, let's see if let's explore this sample characteristics and come back to it. We still have some time, some more things to cover. So so we will download this series matrix file, and that is going to, for example, open up like this. So we have two files, one file or a bunch of folder, which gives us expression values measured for each and every one of these samples. Yeah. Okay. This page link, this is okay. I will get it. Okay, it's here. You don't have to download this now, because I have done the download and I have uploaded the file in GitHub or in shareable link. So that's what we are going to use. But if you want to explore the data page, and this is the publication, you're free to do so here. Okay, so I have downloaded this file. Let's go to the file and take a look at what this file actually shows us. So if you open this file, for example, in Excel or in numbers, so it's going to open up like this, not like this. Yes, like this. So here you will find the different characteristics of the sample that is being measured. And yeah, so here, for example, did it stop abruptly? Okay, I think I have to open this in Excel. Sample characteristics, where is this? Yes, perfect. If you have R installed in your computer, you can use RStudio to to install and then follow along. That's going to be much quicker because maybe many of us are trying to access Google's server, so it is not actually very responsive right now. Okay, can you paste the error sign, Rabbi? Okay, I think I should open this up in Geo accession. This one is here. Installation complete now. Great, very good. So we will come back to that. If you open this in Excel, my point is you will find the sample characteristics here, and let's open this up in Excel. That's going to be so is the installation complete for many of you? One complete, that's great. Okay, here. So here, for example, this is what we are interested in. Let's see here. We have not only the characteristics like which sample comes from which tissue here, and whether this sample is a cancer sample or is it a normal sample, at what stage this cancer or tumor tissue is. So all of these information is available in sample characteristics. So you can download this, and then we can make use of this data also to understand or to associate the phenotypical data as this one with the expression data that we are going to download. As I mentioned, if you find a data with EBI repository, you can actually use that repository to download more visualization friendly data. If you click on this one, and you will see, for example, let it load. Anybody else has gotten their installation complete? Yeah, so this is the page that you will get, and you will have, for example, data from here. I'll give you the link. Right now, let me copy and paste the link. Experiments, yeah. Yes, for example, this is the experimental data, and then, yes, so you can access the expression atlas experiment from here, and we get to that actually from this link. I'll show you where this is. Yeah, so here you get, you can download the experimental design or the sample characteristics output, and the same or similar table, but in a much user-friendly way. So you can download this from here, and you will find these resources. Okay. I'm going to give you this file in a different link or different link. Yeah. Yeah. So, and then, for example, in this downloads, you will also find all raw counts for this experiment. So that also you can download. So these two files, if you download this, this is what you will get. For example, all raw counts, and the same file or a similar different organization for the file that you downloaded from NCBI, right? So this is another experimental design, that file, that both of these we can download from here. So I'll show you what these two files will look like, right? And these two files will look like the one that we want to get our NCBI files processed to a level where we can load it up in R and then start analyzing this data. So this is the experimental design. Installation is done. Very good. Congratulations. So this is the experimental design file, which you will open up, which you can open it up, and then this is what it will open up into. It is the same. So this is much more user-friendly because the organization is not horizontal. It's in a tabular format. So you will find the IDs and the read IDs or files, and their sample IDs and sample characteristics, like which ones are primary tumor, which ones are metastatic, and etc. And other characteristics, like, for example, the stages of the cancer. And yeah, you can use mainly more sample characteristics, like, right, where this is from colon, or from liver, where it is metastasized to, or whether it's a normal sample. So this is what we will use. And for example, your tabulated file will look like this. The raw counts that we have downloaded from the EM tab or EBI will actually look like this. Let's open it up. I think I have opened this already. Yeah. Installation, is anybody else gotten their installation complete? I think I have this. Very good. Excellent. So you can follow the NCBI link from the publication that I pasted, but you don't have to use that to download data, because I'm going to show you or give you this data that I have downloaded, and then made it available for you from our GitHub account, right? So this is, yeah, so this is what you will get, right? And this is what, this is how the data, this is the format of the data that we will also obtain after you run RNA-seq pipeline, for example, in the Taubot Bioinfo platform from the FASTQ files that you can get access to in the NCBI repository. So that is there. So this has gene IDs on the rows, and this goes for example, if I can, yes, all the way to 58,000 transcripts, and yeah, their gene names also involved in this one. And then for each one of their samples, their raw counts estimated, and we will get their raw counts, right? And this is what we will analyze today. So both of these files are, I have made it available in the GitHub link, so you can use that as I paste along. So after this is complete for you, and you open up another code by pressing this plus, right? Insert code below, press this, you will find a small box like this, and in that box you are going to paste this. This is the file that we are going to load, the design file and the raw count file. Both of them, you are going to paste it like this. No, sorry, don't select and paste. Yes, so that's what you will get, and it's, hopefully I can paste everything in this one. Yes, okay. Can you copy this from the chat and then paste it in the in another code block, as I have done? Can you do that? And let me also do it along with you. After you have pasted this, then you will find this small run the cell option and run it. And these two are the files that I have downloaded from this EBI repository, and then just done nothing, put it in the GitHub, and I'm giving it, sharing it with you. So that is easier that way. The power of NCBI, maybe sometimes we might have to pre-process these files to a required format to perform some of the analysis. But NCBI repository has links from this one data page to the FASTQ files repository, and from this one data page to all the other associated options. Like you can even find all the publications there. And yeah, so so that is one hub from where you can get everything associated with this one resources. So sometimes we can tolerate a certain level of requirements to reorganize the data as we get along, right? So once, if the run is complete, let's check what we have loaded. So what did we actually do? Here in the first part, we have installed the libraries, which took forever, and then we have loaded the required libraries. And then here we have just loaded the sample, like the expression table into a data frame called raw.counts, and then we have just another data frame called design will load the experimental design into themselves. So as I told you, as we discussed, so this read.table function requires the path of the file to be mentioned, and then followed by some characteristics of the file. What are the characteristics of the file? I have to mention whether this file has a header or not. Some of the files might not have header, and we have header information in the first row of the file, which tells what each column means to us. So I should tell, or I should mention it to our load.data function that yes, I have header present in my data, and the separator that separates each and every column in my data is a tab. So how do I know that it is a tab? Right? I can open it up in Excel, and if you open this up in Excel, it will show you while importing it that the character that separates the field is tab, right? That's it. It'll show you there in the import data option. Or for sometimes you can observe that the output or the extension of the file is TSV, which is tab separated version or tab separated format. So that we know. Okay, then if it is complete, when it is complete, open another code like this, insert a code like this, press it up like this. So you will find a code like this here, and in here mention or type head of design, and then run this. Copy this, and then please run this. You can, if you don't want to copy and paste, you can run it along with me. So I'm just going to, for example, explore the first few lines of the design data frame I have loaded from this GitHub link or from this file. This is exactly the file that we opened in Numbers or Excel to look through their organization. That's what we need. We just need to make sure that everything is read in a way that we want, like we don't have if for example we don't we only need, for example, the run information or the sample IDs names and their biopsy site or characteristics biopsy site. Okay, that's good. And then if this is done, if you are here, then open up another code along with me, and then now we will find or peek into the expression table, right, that we have named it as raw counts. Right? Head of raw counts. Head would mean display the first few lines of my data frame. And after you have done that, can you see this information? Yes. Okay. Do you want me to? Yeah. Okay. Everybody are here. So congratulations, you have loaded the file into R's environment. Now let's proceed in full speed to analyze this file. So what are we going to do? For example, this is what I have. We have loaded in R's environment, right? Instead of head, if you type just simply design, which is the name of our data frame, and then press, it's going to display everything. So why do I display everything? I just need to find out what is the comparison that I want to set up. I have three types of tissue, right? One is tumor tissue, right? And then the next is normal tissue, and the next is colorectal tissue. So I am going to be interested in comparing tumor versus normal. So that will become part of my comparison that I'm going to set up. So for that, you can actually find out from this, or for example, from opening this file in a numbers format, we will see or we observe, right, from the first sample to 18, right? You remember it started from 2. So from the first sample to 18th sample, we have tumor tissue. And from a 19th sample till 38th or 37th sample, we have normal tissue. So that information we need. And we have several ways of separating which of these IDs or file IDs belongs to tumor and normal. But let's take it really slow, and you will learn other ways in the during the session or during the program. So that's what I'm going to mention here. So look, I am interested in the design section of the design file, which compares or which has information for example, primary versus normal. So that is up to 37th row, right? That's what I found out. So that's what I'm mentioning here. Let me close this. That's what I'm mentioning here, right? Design equals 1 to 37. So I'm copying this, right? Just copy this, and then I will, the next line, open up another code, and then press this, and then put it. So run this, right? I'm just pre-processing the design or the sample characteristics file to the required one. So if you run this, you will not get any output, but run this. And then based on the count that I did, or I have 18 cancer files or cancer samples and 90 normal samples, right? That's what I'm going to create 2 labels for my cancer and normal tissues, right? And yeah, this function is going to repeat this one word this many times. So for example, to this, just to demonstrate what it actually does, this doesn't give you any output, Rabbi, don't worry. We are just overwriting the design data frame with the subset of the design, so it's not going to give you an output. So for example, if I do this, it's just going to create cancer, cancer, cancer 18 times. And similarly, if you do this, this is to, for example, this function is to create a label for our sample, just like just like the sample characteristics that you find. So we need this because we have to prepare, for example, the analysis design file that the differential gene expression analysis will take. So take this also, that is what this is going to do. And I'm going to open up next code block, or even below this, don't do this, open up next code block and run this, right? And run this. This also will not give you any output. Please do that. So let me also catch up with you. So this I wanted you to do first. Okay. And then here I wanted you to do this one. And this is also not going to give me any output. That's fine. And then I'm going to, for example, from this raw counts, right? From this raw counts, I'm going to extract select the variable of interest, gene names, and for example, the design and run. So where is this one? Okay, here is this one. Okay. Raw counts, design, run. Okay, what? Okay, this is what this one. Select the variables of interest in data and add gene names. That is what this is going to run. Use. So we are going to take this option. Let me run this one. Yeah. So this run information, right? This run column is going to get added to, for example, gene name. I don't actually quite get this. What this actually does. Wait a second. Let me take this and explore this further. Okay, yeah. Gene name and design run is going to be here. Okay. Okay. So it is going to, for example, it has to, yeah. So what did this one actually do? What this one actually does is, it's in a convoluted way that I've written it. Okay. Okay. So what this one actually does is, this is going to subset your data set for only the first 38, right? Yeah, first 37 comparisons that we are setting up, right? So if you check this raw count information, so it has 56 columns, as we have all the samples and their expression values measured. So, but we don't want everything. We want only primary tumor and normal, as we have subsetted here, right? So that's what this is going to do. This is going to, for example, load up the gene table, this expression table, raw counts, and then subset only those samples for which I'm interested in. So let's quickly do this and run this. This is also not going to produce any result, right? And run that, and then okay. Once it is actually run, and then we are going to leave out, for example, for the analysis purposes, the gene names inclusion or information from our table. So after this, if you are there, now I want you to open up another code and, for example, do this, right? Head. If you do this, all you will observe is a data frame which has 37 columns, matching our design, our comparison design, and 6 because we have added only head first few lines of the code. So are you there yet? Yes, excellent. So that's everybody's progressing so very well. So this is what we need. So all of these 3 blocks is going to first take a subset of your comparison, because our data has 3 comparisons. We want to take a subset of our comparison, define those comparisons, right? And then make the expression table also, filter the expression table also based on the data that we are going to compare, based on the sample sets that we are going to compare. So that's what we have done so far. And then we are going to create a metadata data frame, right? This is also required, right? This is going to be in the next one. So open up error. What is the error? Let me check the error once, but others open up another code from here. I'm going to open up another code from here and then paste this information. This is also not going to give you any output, and then run this. Okay, what is the error that you get, Ume? And please paste it there. And is everybody here finished up this step? After you are done this step, excellent. So open up another code and then type metadata, right, with uppercase. And then enter, right? We should see an output like this. I'll come back to that one. You should see an output like this. So what have we done so far? After loading up the data, we have pre-processed or prepared the input gene expression table to include only the samples of comparison, and prepared or prepared the comparison list from the sample characteristics or experimental design into a separate data frame called metadata. So everybody here, they got data frame 37 into 2. You said object gene table not available. So did you run this? Did you run this particular portion without any problem? Ume, I think you have problems in this particular portion, the one that I have blocked. You did not run this probably before the gene table. So if you run that, then you will get this information done, right? Follow this along. Yes, I will do that. This has to be run before the head gene table and followed by another block and this, and then followed by, for example, a block where we are going to subset or create a metadata data frame, and then followed by printing out the metadata, right? It'll take only another 5 more minutes or 5 to 7 minutes, depending on if everybody is here. This is the flurry of pastes is specific to Ume Hania Irfan, who got problems when she tried to execute this head gene table. So is everybody here after you have printed out metadata? Let's move along. Are you here? So now we are going to perform differential gene expression analysis. Very good. So this is what we are going to perform, and open up another code and paste this and run it again. So now you got your everybody's pro. You don't have to wait for my explanation what to do. So paste this and run. You are going to see, for example, outputs like this, like what the differential gene expression analysis is actually performing. You observe here, we are giving, there are 2 inputs or 3 inputs for creating this data from matrix for the differential gene expression analysis using DSEC library. The first one is the gene table that we prepared. The second one is the metadata that we prepared. Both of these we filtered from the original data. And the third is the type, which is actually the name of this column, which tells which sample is cancer and which sample is normal, or which comparison that we are actually performing, to be exact. And then followed by, this is preparing the data matrix, followed by performing the DSEC analysis. So it's going to run something like, it's going to estimate the gene dispersion for your samples. It's going to, for example, understand how the mean of the genes per sample and how the dispersion is actually related. And based on this, this is going to fit a GLM model to identify the baseline gene expression of housekeeping genes in the cancer samples and normal samples. And then it's going to find differentially expressed genes for cancer versus normal. Why it is not working for you, Hervé? What is the what's the output that you get? What are you getting as an output? Is everybody else were able to finish this? Yes, yes, okay. Okay, then we move along. So what do we do next? Yes. Yeah. So what do we do next? We are actually going to, for example, estimate how many genes are upregulated and how many genes are downregulated. So that is what we are going to do in this specific code block. So can you copy this and okay, can you run this in the next associated one? So after this, what do you get? Can you let me know? Do you get this output? Yes, very good. So what have we just done? We have, out of 40,000 odd gene expression, right? We have estimated which ones are significantly differentially expressed. And from our analysis, we have set a very extremely stringent p-value, like that's like 1 in a million, right? That's really extremely stringent p-value. And with some correction to the adjustment method. Why we set this p-value and why we use this correction will be explained a lot in the program, right? So, and now we also identified something like 121 genes that are upregulated and only 30 genes that are downregulated, right? That's what we find from our analysis and so far. So these statistics are very good for displaying, but how to make this presentable in a for example, plot-like view? So for that, we can use a volcano plot option, which we installed earlier. So open it up and paste this. And let me know, do you get a graph or do you get a plot? Then this one did not run for you, Hervé. Then yeah, we will share this collab with you. Try to follow right from the beginning. Then it should not be a problem for you. Okay. So did you get this output? Can you see a plot like this? Yes. Okay. Excellent. Thank you. So what did we just identify? We have identified genes that are significantly differentially expressed. And for example, You're getting error from metadata. Yeah, you are not preparing some metadata, right? Both of you can follow this collab when we share it with you, right? So what did we just identify? We have identified based on log fold change and based on the p-adjusted value that we have calculated, what are the genes that are significantly differentially expressed, right? And for example, we can also have another version of this plot where we are going to set some stringent cutoffs, right? So that we don't display everything like a beehive. So this is very difficult to interpret what is there and what these different dots mean. So these different dots are the genes that we have identified them as differentially expressed. And this is a better volcano plot where we are going to display only few genes that are differentially expressed. Right? And this is also based on some cutoff that we have set up. So what cutoff to set up? How are we going to set up this cutoff? What do they mean? What does the p-adjusted value mean? Like p cutoff mean, and what does the fold change cutoff mean? And how are we going to, for example, display come from here to here, where we are displaying everything based on, for example, our very lenient cutoff, right? Here, based on the stringent cutoff, we are displaying only few of the genes. Yeah, and yeah, I'm done, Ojasvi. So this analysis and what's the biological implications of this, we can discuss much more detailed in program, but this is the basis of getting to those 19 genes that are identified as being significant in the tumor progression for colorectal cancer based on that paper, right? We are also somewhere close to that, right? How many genes? Here we get something like very close to, for example, 50 genes, less than 50 genes. So this is, yeah. So everybody there, did you get this second plot? Excellent. Thank you very much. And if you have gotten the second plot, this is how you can take a data from publicly available resources and follow along some of the comparative analysis. So this is only differential gene expression analysis, only one lesson from the R program that we are going to cover. So we will do what to do before this analysis and how to perform pre-processing, and what to cover after this analysis if this is not able to identify differentially expressed genes, and what will we do next this one. So with that, thank you for the patience, and we are going to I'm going to hand this over to Sparsh, I believe.
Yes, Dr. Rag. So it's me, yeah. Thank you, Dr. Raghavendran, for such extensive hands-on session. And so I'll be quickly explaining the registration part for all the participants who have joined and want to further enroll in this program and to learn more hands-on as well as conceptual and theoretical skills. So let me share my screen. Yeah, definitely everyone will get a recording after the session is over. We'll be sharing the insights of the session. Also, we are sharing the YouTube link here on the chat itself. Maybe Ojasvi can share the link. So Ojasvi has shared the link already. So please copy this link and you can go through with the process and everything in detail afterwards. So, okay. So could you please confirm if my screen is visible?
Yes, Parsh, it is visible.
Yes, it is visible.
Thank you, sir. Thank you, Shigori. So today was the free webinar for the introduction to bioinformatics in R, or getting started with R, and the program is going to commence from 5th of the December. And it's like a 3-month long duration, depending upon which particular participant is interested in which tenure. So as we all know that the data is growing, and with time, everyone, if you are seeking a job, or if you want to do further research, so coding has become like a kind of must have skill for a bioinformatician or for a normal biologist also. So with this particular program, we will be enabling the students, the researchers, the biologists, the faculty, everyone, so that they can gain both the conceptual as well as the practical skills related to the R with bioinformatics. You will be leveraging to perform in-depth statistical analysis through this program, visualization and annotation, and much more things. So now as I scroll down, this is the program overview. This is the program curriculum, which you can see on my screen. So we will be starting from like the basic introduction to getting started with bioinformatics and R, and transiting you all from beginners to intermediate level, and then in the end at the advanced level we'll be discussing about how you can draft your research question, your research proposal, and further work on your research project. So the very first session, which will be like, which we'll be conducting on December 5th, it will include topics like introduction to big data in bioinformatics, introduction to bioinformatic languages using the code playground R, loading data in R, etc. So as you can see, these are the titles and the topics covered, and also the associated online resources have been mentioned here. The second is working with sequences in R, annotation of genomic variants in R, transcriptomics, data processing and visualization in R. Then also we are covering statistical aspects, differential gene expression, machine learning aspects, metagenomic analysis in R, and so on. So we will be like covering three different multi-omics study domain, that is genomics, transcriptomics, metagenomics, with machine learning and statistical aspects. So as I move further, there are three different tenures in which you can enroll further, and those are of for 45 days, 60 days, and 90 days. And for those who are from, obviously we have participants across the globe. So anyone who wants to see the pricing for the three different tenures, they can accordingly choose the map where they are residing. For example, if someone is residing in US, so they can choose it for US, US. Then if someone is in India, they can choose accordingly to the currency, that's INR, then in euros, then in Brazil, etc. And if someone is living in Nigeria or African parts, they can choose the Naira currency. So for now, I have displayed the payment link in INR, and if you want, you can change it to USD also, to Nairas also, to Euros also, etc. So now I'll be telling about what are the common resources which one would have access to if they'll enroll in this particular program. So the basic access is online tutorials. You will be having access to the Omics Logic Learn portal. Then you will be having access to all the example research projects on Omics Logic Learn portal, then the coding sessions, the session recordings, obviously the scripts also for our then online mentor support, online technical support. And once the program is over, the program is finished, you will receive a certificate of completion also, and then you will have access to some example cloud pipelines on our T-Bioinfo platform. So these are the common resources which one would have for all the three tenures. Now, what's the difference between the three tenure is, in 45 days, you will have access to group discussions, live Q&A sessions, or live sessions. In 60 days and 90 days tenure, you have two different opportunities. In 60 days tenure, you will be having access to one-on-one mentor session. For example, you are going through with some of your session, or you are going through with some of your coursework or lesson on Omics Logic Learn portal, and you came across any question or query. So we can fix a one-on-one mentor session of yours with the mentor, and you can get your queries clarified. It would be a kind of a 15 to 20 minutes exploratory meet with the mentor. And the second important feature, or the most you can say the opportunity which you'll have, is that you have an opportunity to draft a research proposal and present it to our expert mentor for the valuable feedback and evaluation. Then for the 90 days tenure with one-on-one mentor sessions, you have an opportunity to draft a complete research project and present it to the mentor. And also you can further work on that research project and get it published in a good peer-reviewed journal. So this was the resources which you'll have access to when you enroll for this particular program. And now we are providing scholarship to everyone, like early bird kind of scholarship to everyone. And I guess Shri Gauri or Ujasvi can share. Ujasvi has already shared the coupon code. So I'll just quickly show you how you can proceed with the payment checkout also. So for example, if I'm interested in 45 days tenure, I'll click on the buy now option, and just a moment, it's loading. Yeah. So as you can see that here they're asking for if we have any coupon code. So the coupon code is take, all in caps, TAKE, take 20, and then apply. So the coupon code is successfully applied. You can see here the cost has been decreased. Earlier, the actual cost was in INR 7,500, and now it is decreased to 6,000. So it's a 1,500 discount, a 20% scholarship for all the Indian contacts. And if, for example, if I'll do it for like USD people who are living in US or so, I'll just show you a glimpse. So now you can see the pricing has been changed. For 45 days, it's $150. Then for 60 days, it's $300, and for 90 days, it's $450. So now if I'll click again on buy now, I can simply write here, type take 20, and then apply. And as you can see that here, the pricing has been changed to $120 from $150. So a reduction of $30 is there, and you can further proceed with maybe Stripe payment method or PayPal payment method after filling out the user details. So this is how the payment process is so easy, and this coupon code is applicable till 5th of the December. So please, like, if you are interested and you want to avail this opportunity, and so please go and start enrolling yourself. Now one more thing, if anyone who is like landing up to this particular page, it's our landing page of the program, and we have also incorporated the payment link here on the landing page also. So if anyone is interested in any of the tenure, they can simply click on the tenure and again type just the coupon code, that is take 20, and proceed with the payment option. Add discount code, and you can simply write here take 20, apply. I guess there is, yeah, discount code applied. See, the total has now been changed, that is $120. So this is how you can proceed with the payment checkout through our both of the pages, that is program page as well as the landing page. And so this was all about the resources, the registration, and the scholarship part. If anyone is having any query, they can ping me directly on my email address, that is marketing@omixlogic. Thank you, Shri Gauri. So Shri Gauri has already shared the details. So it's marketing@omixlogic.com. Even the official number, that is +91 987613412. It's active on WhatsApp also, so we can directly get in touch and we can have a telephonic conversation if you feel so that you have some queries and you want to get it resolved. So this was all about the registration part and the payment process and the scholarship. So if anyone is having any kind of query related to registration, please feel free to ping in the chat box and I'll be there to answer your queries. Thank you. Over to you, Shri Gauri.
All right. So I guess the participants have no further queries and we are good to end today's meeting. And if the participants are having any other queries, you can feel free to reach out to the contact details that I've shared in the chat. And there is one query: any further discounts for LMIC?
So it's low income country, if I'm not wrong. We can definitely like we have a scholarship form for like if you further want more scholarship. So you can mail us at marketing@omicslogic.com, and then we can provide you with the scholarship form, and then we can further discuss it with the internal team and get back to you. So definitely we can help you out with more scholarship or discounts.
And then we have another query: how long the session will be for courses? Is it daily basis?
No, it's not daily based. We have set particular dates when the session would be conducted, and like the sessions, the program is hybrid. Some of the sessions would be live, and some of the sessions would be asynchronous, depending upon if the hands-on, like if we are doing hands-on and it's the practical part is more, so the sessions would be live, obviously. And when if something conceptual needs to be get covered, we can we can maybe send you asynchronously, and you can go through, and then maybe when we conduct the live Q&A sessions or through online mentor support, you can get your queries clarified.
All right. So I guess that's all the queries. And along the way, if you're having any queries, please feel free to reach out. And with that, I hope you were able to learn something new today from today's webinar. And I look forward to seeing you all for the our program as well. And with that, thank you everyone for joining today's webinar, and have a great day.
Thank you, everyone.